Hello there guys, welcome back or welcome if you're joining us for the first time. We're back. And in today's video we're looking at taxonomy of bacteria. Clinically, bacteria are classified based on their gram staining because of their cell wall, their shape or morphology, and their metabolism, how they react to oxygen and how they get their energy. Let's begin with gram staining. Now the gram staining procedure has four steps. The first step is staining with crystal violet, which turns everything blue, both the gram positive and gram negative. Next, we add iodine, which will basically trap this blue staining in gram positive bacteria. And then we have alcohol wash. For negative bacteria, it will wash out the blue stain. Then we have saffron dye, which we then color everything red. But since the gram positive still trap the blue stain, they remain blue. But the gram negative now becomes stained red. So this is the structure of the gram positive cell wall with a thick peptidoglycan layer and the gram negative with its own thin layer. This peptidoglycan layer is not only important for the gram staining, but also very important for antimicrobial therapy using penicillin. Now, penicillin acts on the enzyme that cross-links peptidoglycan. So, if you give penicillin and it gets to peptidoglycan, it makes that bond weaker because it acts on that enzyme. And as such, a cell that has a thick peptidoglycan layer, like the gram-positive bacteria, will easily absorb water and then burst because its cell was no longer rigid. Now, this same thing theoretically can happen to the gram-negative bacteria, but they have a defense. Just over their thin peptidoglycan layer, gram-negative bacteria have an outer membrane that contains porins that allow only small nutrient molecules to pass through. So complex molecules like peptidoglycan can go through, and as such, their cell wall is protected. Now, still on the gram-negative bacteria, on their outer cell membrane, they have lipopolysaccharides, which have three layers: an outer, middle, and inner layer. The outer layer is antigenic in nature and differs from bacteria to bacteria. They have a middle polysaccharide core and an inner layer which contains lipid A, which is an endotoxin. So now, when these gram-negative bacteria are destroyed, it releases this endotoxin throughout your bloodstream. And when your white blood cells sees this, it triggers an overactive inflammatory process, which causes high fever and other things you see when you have a gram-negative bacterial infection.